Today, we're diving into something called targeted protein degradation. Now, this is a totally revolutionary approach in medicine. We're not just talking about blocking a disease, we're talking about aiming to completely delete it from our cells. Okay, think about it like this. For decades, right, medicine has been all about blocking the proteins that make us sick. But what if we could just tell our body to, I don't know, throw those bad proteins in the trash, just get rid of them, erase them completely? That's the incredibly powerful idea we're going to dig into. So to really get why this is such a huge game changer, we first have to talk about a massive challenge in modern medicine. It's what scientists call the undruggable problem. Most drugs you've ever taken, they work on a really simple principle, the classic key in a lock. The drug is the key, and it's designed to fit perfectly into a specific part of a protein, that's the lock, and just jam it up, stop it from working. And look, it's an incredibly effective strategy when it works. But here's the massive catch. A huge number of proteins that cause diseases, they just don't have a good lock for a drug key to fit into. And for others, it's not even about what they do, it's just the fact that they're there that's the problem. These targets, man, they're implicated in everything from cancers to neurodegenerative diseases. And for a long time, they've been considered totally undruggable. So it turns out the solution to this problem wasn't something we had to invent from scratch. We just had to look inside our very own cells and find nature's own super efficient cleanup crew. See, every single one of your cells has this incredible piece of machinery inside called the ubiquitin proteasome system, or UPS for short. Just think of it like a quality control manager and a recycling center all rolled into one. Its whole job is to constantly seek out and destroy old, damaged, or just plain unneeded proteins. Okay, I know this diagram looks a little complicated, but what you're seeing is a beautiful process that's happening inside your body literally right now. In super simple terms, on the left, you've got this system of enzymes, E1, E2, and E3, and they're all working together to grab this tiny tag called ubiquitin. Then they stick that tag onto a target protein, basically marking it for destruction. And that destruction is carried out by the proteasome, which is kind of like the cell's paper shredder. This is the body's natural way of cleaning house. It really all boils down to 3 simple steps. First up, tagging. An enzyme called an E3 ligase acts as the tagger, and it slops a small protein called ubiquitin onto the target. You can think of it like a molecular kick-me sign. Next, more ubiquitin tags get added on, forming a chain that's basically an unmistakable signal for disposal. And finally, the proteasome, the cell's shredder. It recognizes that chain, grabs the protein, and boom, completely dismantles it. Okay, so if our cells already have this amazing disposal system, could we hijack it? Could we use it to get rid of any protein we want? Well, the answer is a resounding yes. And the molecules that do this hijacking, they're called degraders. And this is where the genius of targeted protein degradation really shines. Scientists have engineered these molecules that don't block the target protein at all. No. Instead, they act as a kind of molecular bridge. They force an introduction between the disease-causing protein and the cell's E3 ligase, the official tagger for the garbage disposal. And there are 2 really clever ways to do this. The first strategy uses something called PROTACs. Think of them as molecular matchmakers. They're these 2-headed molecules with a little linker in between. One head grabs the problem protein, the other head grabs the E3 ligase, and it physically yanks them together. The second strategy is molecular glues. Now, these are smaller molecules that act more like superglue. They bind to the E3 ligase and actually change its surface, making it sticky for a specific target protein that it would normally just ignore. And this slide right here shows the difference perfectly. On the left, you can see that PROTAC acting as that physical bridge, right? It's forcing the 2 proteins together. But on the right, the molecular glue isn't a bridge at all. It's actually reshaping the E3 ligase to create a brand new docking site. So 2 totally different tactics, but they both have the same ultimate goal, destruction. Now, this technology, it sounds incredibly futuristic, right? But the definitive proof, the thing that showed it could actually work in humans, it didn't come from some fancy modern lab. Nope, it came from a decades-long investigation into one of medicine's most infamous and tragic drugs. So back in the 1950s, the drug thalidomide caused this global tragedy, leading to thousands of severe birth defects. But then, decades later, scientists were shocked to find it was a powerful treatment for the blood cancer multiple myeloma. And for years, nobody could figure out why. The mystery wasn't solved until 2010. And the discovery was just stunning. Thalidomide didn't work by blocking anything. It was a molecular glue. It was tricking the body into destroying the very proteins that were keeping cancer alive. This discovery, as one scientific paper put it, provided clinical proof of concept and established the efficacy and safety of molecular glues. It basically validated the entire field overnight. Let's take a closer look at what's happening at the molecular level. Right here, you can see that thalidomide-like molecule, shown in yellow, acting as the glue. It's perfectly nestled between the cancer-causing target protein, that's the TL1, and the cell's disposal machinery. It is literally sticking them together, condemning that TL protein straight to the shredder. Amazing. So, unraveling the thalidomide mystery wasn't just about solving an old case. It was like firing the starting gun for a race to build a whole new class of medicines. We are now truly in a new era for drugs. And look, this is not just some theory anymore. Building directly on that accidental breakthrough, there are now dozens of new, rationally designed protein degraders in clinical trials as we speak, targeting diseases that were once thought to be completely untreatable. And the potential applications are just breathtaking. I mean, we're talking about entirely new ways to fight cancers, to clear out the toxic proteins that cause neurodegenerative diseases like Alzheimer's and Parkinson's, to tackle autoimmune disorders, and maybe even to eliminate viral proteins from our bodies for good. Ultimately, what's happened is we've cracked a fundamental code. We are literally learning to speak our cell's native language of cleanup and repair. And that leaves us with a really profound question for the future. With this powerful new toolkit, with the ability to target almost any protein for destruction, what diseases are we going to choose to erase first?